Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about the genetic code. The genetic code is a set of three-letter combination of nucleotides called as codons, each of which correspond to a specific amino acid or to the stop signal. The genetic code is a non-overlapping code in which each amino acid plus the polypeptide initiation and termination are specified by RNA codons that are composed of three nucleotides. The concept of codon was first described by Francis Crick and his colleagues in the year 1961. Friends, two experiments have contributed immensely to the deciphering the genetic code. These two different experiments are in vitro translation experiment by using synthetic mRNA. Second experiment is trinucleotide binding assay. By combining the result of these two experiments, several scientists, namely Marshall Nirenberg, Severo Ochoa, Hargobind Khurana, Philip Leader and his colleagues, they succeeded in deciphering the genetic code. Out of these scientists, Marshall Nirenberg and Hargobind Khurana, they shared the 1968 Nobel Prize in Physiology or Medicine for their work on genetic code, along with Robert Holle, who determined the complete nucleotide sequence of alanine tRNA in yeast. Previously, Severo Ochoa already had received the Nobel Prize in Physiology or Medicine in 1959 for the discovery of RNA polymerase. The genetic code is composed of 64 codons. Out of 64 codons, 61 codons specify various amino acids, and each codon is a triplet code. That means each codon is composed of 3 nitrogenous bases. So out of 64 codons, 61 codons specify various amino acids, and remaining 3 codons are called as nonsense codon or termination codon. These 3 termination codons are UAA, UAG and UGA. This termination codons causes the termination of protein synthesis. Now we shall know about the properties of the genetic code. The genetic code is composed of nucleotide triplets. It is non-overlapping. The genetic code is comma-free and is degenerate. The genetic code is ordered. And the genetic code is having start codons and stop codons. And the genetic code is nearly universal. These are the properties of the genetic code. Now we shall discuss about some of the properties of the genetic code in detail. The genetic code has start codons and stop codons. Start codons are also known as initiation codons, and the stop codons are known as termination codon. In both prokaryotes and eukaryotes, AUG is the initiation codon. It is used to initiate polypeptide chain. Rarely GUG is used as the initiation codon, but in rare cases. In both prokaryotes and eukaryotes, in both cases, the initiation codon is recognized by initiator tRNA. In prokaryote, the initiator tRNA is formyl methionine tRNA. Formyl methionine tRNA is the initiator tRNA in prokaryotes, whereas in eukaryote, initiator methionine tRNA is the initiator tRNA. The internal AUG codons are recognized by another type of tRNA, that is methionine tRNA. This methionine tRNA is different from this methionine tRNA, whereas the internal GUG codon is recognized by valine tRNA. In the genetic code, there are three stop codons or termination codons. These three stop codons are UAA, UAG and UGA. These three stop codons specify polypeptide chain termination. These three stop codons or termination codons are recognized by specific protein known as release factors. In prokaryotes, there are two types of release factors, RF1 and RF2. RF1 terminate polypeptide chain synthesis in response to codons UAA and UAG, whereas the release factor 2 terminate protein synthesis at the codon UAA and UGA. In eukaryotes, there is a single release factor that can recognize all the three stop codons. One of the important property of the genetic code is that the code is degenerate and adult. What does it mean? Friends, out of 20 standard amino acids, methionine and tryptophan are the two amino acids that are specified by a single codon each, whereas remaining 18 amino acids are specified by more than one codon. This is the degeneracy of the genetic code. Three amino acids, they are leucine, serine and arginine. They are specified by 6 codon each, whereas isoleucine is another amino acid specified by 3 codons. Remaining amino acids are specified by either 2 or 4 codons. The degeneracy of the genetic code is not at random. The degeneracy is highly ordered. What does it mean? In most cases, multiple codons specifying an amino acid differ by only one base, that is the third base or the 3' base. The degeneracy is of two types, partial degeneracy and complete degeneracy. In partial degeneracy, the third base or the 3' base can be either of the two purines, that mean adenine or guanine, or the 3' base can be either of the pyrimidine, that is either uracil or cytosine. But in partial degeneracy, changing the nitrogenous base from a purine to a pyrimidine will also change the amino acid. This is about partial degeneracy. What is complete degeneracy? In case of complete degeneracy, any of the 4 nitrogenous base can be present at the 3' position. Still, the code will specify the same amino acid. That is the complete degeneracy. Another property of the genetic code is that the code is non-overlapping and comma-free. What is the meaning of non-overlapping genetic code? Non-overlapping means each nucleotide is the part of a single codon, and the genetic code is comma-free or comma-less. That means the triplet codes are arranged one after another without any gap or space. So the genetic code is comma-less. The most important property of the genetic code is its universality. With minor exception, the codons have the same meaning in all species. The most important exceptions to the universality of the genetic code occur in the mitochondria of mammals, yeast, and several other species. Mitochondria are the semi-autonomous organelle. They have their own chromosome and protein synthesizing machinery. Although the cytoplasmic and mitochondrial system are similar, still there are some differences. In the mitochondria of humans and mammals, UGA is the codon that specifies tryptophan rather than chain termination. In the cytoplasmic system, UGA is the termination codon. But in the mitochondria of human and mammals, UGA specifies tryptophan rather than chain termination. Another codon is AUA. AUA is the codon that specifies methionine rather than isoleucine. Two codons, one is AGA and AGG. These are the termination codons in case of mitochondrial system. Whereas in cytoplasmic system, AGA and AGG are the codons that specify arginine. The remaining 60 codons have the same meaning in the nucleus and the mitochondria. Friends, this is about the genetic code. If you have any doubt regarding any of the topic related to biological science, then you please inform me through comments so that I can prepare a video and it will be uploaded in my YouTube channel as early as possible. Thank you very much, friends. See you again. Bye bye.